Quantifying gene-centered content in Illumina's correlation engine. In this demo, we'll briefly examine ways to use correlation engine to interpret RNA-seq and other data types. Then, in the context of understanding genes that may be among a set of, for instance, differentially expressed genes, we'll start with a set of gene identifiers to quantify and compare the available literature-based knowledge in PubMed with the number of significant results when queried across correlation engine's public omics studies. We'll focus on what this may say about the potential for insights from correlation engine, especially for less well-studied genes. Next, we'll hone in on a set of 3 genes from a specific study. And we'll end with the different ways to access gene-centered content in correlation engine. The correlation engine knowledge base can be a great solution for helping you interpret the results of your omics studies. This particular example corresponds to an RNA expression study in which a keratinocyte cell line was treated with hydrogen peroxide and compared to untreated cells. You see that the 3 top regulated genes here, with p-values less than 0.05 and ranked by magnitude of fold change, are MAX16, PAXBP1, and PDS5B. One way we can begin to better understand what's going on in the cells in response to treatment is by using the Pathway Enrichment app. The app finds correlations between the genes differentially expressed in the treatment and those comprising knowledge-based biomarker sets or biogroups. These biogroups can correspond to gene ontology sets or, for instance, mSigDB pathways, as selected here. Or, using the Correlation Engine Curated Studies app, we may query against 189,000 plus processed public omics data signatures or biosets to see in which prior studies many of the same genes were significantly altered with respect to expression or otherwise depending on the omics data type. You may additionally find value by investigating the specific genes that rank among the most differentially expressed that may be new to you. PubMed is a good resource to find literature to help understand what's known about the gene's function already and to explore why it may be over or underexpressed in your particular study. For a gene such as Notch1, the top-ranking gene in this alternate prostate cancer cell line study, if we search for the gene in PubMed, we see over 9,000 matching papers, including reviews to help figure out why Notch1 may be altered in that study and what effect it may be mediating in the cell line. On the other hand, in our keratinocyte cell line study, if we're not already familiar with MAX16, PAXBP1, or PDS5B, the 22 results, 10 results, and 85 results for these genes may not provide enough information to help us understand why the gene's expression may be altered or what effect the altered expression may have on the keratinocytes. In such a case, another means to begin to understand what role a gene may have is to examine where the gene figures significantly among all the omics studies represented in Correlation Engine. We see that a gene such as Notch1 is well represented as a significant finding among over 9,000 correlation engine data signatures. It's less certain whether we'll obtain as many results for genes that are less well studied, such as MAX16, PAXBP1, or PDS5B from the keratinocyte study. To this end, let's examine the representation of genes in PubMed and Correlation Engine. Comparing their representation allows us to understand how the results may vary based on what's already understood about them. It's possible, for instance, that genes that are poorly understood based on their representation in the literature may also be poorly represented as significant results in omics studies. Let's find out. Because content is continuously being added to PubMed and Correlation Engine, note that this analysis was performed in May of 2023. We started with 57,116 human ensemble gene IDs and plotted the number of significant results found in Correlation Engine against the number of PubMed search results. In the initial plot, it's hard to see the full distribution because a few genes score with millions of PubMed hits. Upon further examination, the top 49 are dominated by false positive results by virtue of having names like Anne, Th, and Nov. Let's remove those. Then, examining the genes that have either 0 hits for PubMed or 0 hits for Correlation Engine, we find that most of those correspond to pseudogenes or other non-protein coding genetic elements. If we remove those, 21,589 genes remain. The results for PubMed vary over orders of magnitude, so let's shift to a logarithmic scale and set data points to 75% transparency. With those adjustments, we see that indeed the number of PubMed search results still vary widely, while most Correlation Engine results seem to fall between 1,000 and 10,000 significant results, whether the genes are less well studied or better understood based on the PubMed results. Let's see what a histogram view can tell us. 70% of the 21,000 plus genes have less than 100 PubMed search results, and 34% have only 1 to 9 PubMed search results. In comparison, most of those genes have between 1,000 and 9,999 significant matches to the public omics studies in Correlation Engine. In other words, the open hypothesis approach of omics studies means that there can be more balanced coverage and therefore greater potential for insight for understudied genes, the bulk of genes whose function and roles we still don't fully understand. Another way to summarize is based on the median result. The median number of significant results found for a gene in Correlation Engine is 4,079, and the median number of results in PubMed is 28. Now let's take a look at the results from the keratinocyte study, specifically for MAX16, PAXBP1, and PDS5B. We see 8,403 significant results for MAX16, 8,045 results for PAXBP1, and 8,935 significant results for PDS5B. These are results from the Curated Studies app, but there are multiple other ways to investigate gene function using the QuickView app. For instance, find general information, which includes information about gene ontologies, pathways, and protein families the gene is known to be a member of. Or from the Summary tab, access additional apps such as Body Atlas, Disease Atlas, Pharmacoatlas, and Knockdown Atlas. We already saw that we can see basic information, such as gene ontologies, pathways, and protein families that the gene's a part of in the QuickView general information. From the Body Atlas, find which organ systems, tissues, and cell types the gene is expressed in. Use the Disease Atlas to see in what diseases the gene expression is altered or in which the gene is mutated. The Knockout Atlas allows you to see which gene perturbations affect the gene's expression. And the Pharmacoatlas gives insight into compound treatments which impact gene expression. Finally, the observation that just a small number of genes are overrepresented in the literature has been recognized more broadly for some time. In fact, out of the 20,000 or so human protein coding genes, just 100 account for more than a quarter of the papers tagged by the National Library of Medicine. and the top 10 of these genes are shown on the right. Consult some of these articles to learn more about why some genes are overstudied at the expense of the broader set of genes, the functions of which we still have yet to fully understand. With this in mind, Correlation Engine can be a valuable resource to fill in the gap between what's known about genes from published literature and what can be gained from access to thousands of processed public omics studies. If you don't already have access to Correlation Engine, request a free trial to explore the possibilities. Thank you for watching.